Hi, everyone. Welcome to Microbial Minutes. This is the Microbial Minutes update for February 26, 2018. I'm Julie Wolfe with the American Society for Microbiology, here to talk about what's hot in the microbial sciences. Got a number of updates for us in the last couple of weeks. We're going to talk about a new bird flu, as well as a general update on the flu season. We're going to talk about some infrastructure in our water piping. First, talking about bacteria in water pipes associated with the Flint water crisis, and then talking about the transmission of antimicrobial resistance genes in hospital piping. Then we're going to talk about a new strain of extremely drug-resistant typhoid fever. And finally, ending on a more positive note, talking about a new class of antibiotics found in soil. We're going to look at the original reports of all of these different things. As always, this is a live session. You are welcome to put a comment into the chat box and we will address that, or leave a comment later on if you're watching this not in the live session, and we're happy to answer it that way as well. So let's jump right in and talk about a press release that was released from the government of Hong Kong, which the take-home message is pretty straightforward. There's another new avian influenza strain that has jumped into people. Now, we've covered avian influenza quite a bit in Microbial Minutes. It's something that people, public health workers, epidemiologists, microbiologists worry about quite a bit, jumping into people, and then not only jumping from birds into people, but then becoming transmissible from person to person. Most of the strains that have been characterized so far have only jumped into people, and this includes 3 cases of H7N9. That was a strain that we discussed back in October, talking about ferret transmission of this highly pathogenic H7N9, but it has yet only been able to transmit from birds into people. There have also been some cases of H5N6 and many different cases of H5 outbreaks in birds. Now, remember, the way that we differentiate different influenza strains is by calling them by the different hemagglutinin, or H, and neuraminidase, or N. These are the different proteins that decorate the outside of the influenza particle. And a new strain has been found that has caused an influenza infection in a person, and this is an H7N4, never before seen in people. This happened in a 68-year-old woman. She was admitted to the hospital with very severe disease, but did not die of this disease. It took her a few weeks to recover, and she was discharged having recovered from that influenza. But it's always important to make note when there is a new virus that has been able to transmit from birds into people. And this woman did describe some close contact with live birds, poultry, before the onset of her symptoms, and that is likely the source of her infection. This is something that was widely shared on social media, different platforms, by a number of scientists who follow avian influenza and potential strains that may cause a wider disease in people. And this led to the question of just how many avian influenza strains are there? How many strains of influenza are there? There are 9 different hemagglutinins and 16 different neuraminidase genes that have been identified so far. And so if you take a combination of all of those, there's 144 different subtypes that could potentially cause disease in various different organisms. And we have a nice summary of this in an NPR news story that we'll include in the links below the session window here. Now, let's just update a little bit on the current influenza outbreak. We're in a really severe epidemic year following this red line. This is the 2017 to 2018 year. You can see that it peaked very, very high as the 2009 pandemic year. And this is an important differentiation. A pandemic occurs when a new strain that has not been found in people jumps into people. And that was the case with that H1N1 pandemic strain. The strain that has been infecting most people this year, at least in the United States, is the H3N2. This is something that has been circulating in the population since the late 60s. And so it's not a new virus, but it is the one that has decreased efficacy in the vaccine this year. And So where there's a nice summary of the difference between pandemic and epidemic in a Washington Post story that we'll include a link to. And this efficacy issue is perhaps one of the reasons why flu mist has recently been reapproved. It's the nasal inhalation vaccine. And this was covered by the reporter Helen Branswell, who, if you're interested in influenza, what is really going on in influenza, following her on Twitter is just one way to always be on top of what's going on. But of course, influenza vaccine has to be prepared so far in advance that this is not likely to affect next year's vaccine stocks. It will probably be something that affects subsequent years after that. And that's in part because it takes such a long time and so much infrastructure to gather all of the, in our case, eggs, right? So most of the influenza vaccine is made in eggs, and to have the infrastructure to switch to generating the flu mist is something that will probably take a little bit of time. If you're interested in more influenza factoids, there was a really great episode of This Week in Virology recently called The PFU in Your Chew with Scott Helmsley. It was discussing a number of recent pieces of literature, including looking at how many viral particles we expel just as we speak, not only when we sneeze or cough. And so I have the link on here, and I highly recommend it. It was a good conversation. Moving forward, we're going to talk about two recent articles that talked about the Legionnaires disease outbreak in Flint, Michigan. One is from PNAS and one is from the ASM journal MBio. The PNAS journal the article was titled Assessment of the Legionnaires disease outbreak in Flint, Michigan. And the MBio article was titled Prevalence of Infection Competent Serogroup 6 Legionella pneumophila within premise plumbing in Southeast Michigan. The take-home message from both of these is that the Legionnaires disease outbreak is tied to Flint water quality. So there was a pneumonia outbreak caused by Legionnaires disease that happened simultaneous to issues with the Flint water quality, the infrastructure, and these reports really tie them together. So let's review the timeline of what happened with the Flint infrastructure. In April 2014, the Flint water source was switched from Lake Huron, one of the Great Lakes that surrounds the state of Michigan, to the Flint River. And almost immediately, the residents started to complain about a bad discoloring in their water, a bad smell in their water, and some people who were bathing or washing their hands with this water started to complain of skin rashes. In 2014, there were 45 cases of Legionnaires disease. In a normal year, there are only 12. And between 2014 and 2015, there were nearly 90 cases of this pneumonia. This is a very specific type of pneumonia. It's not transmissible person to person. It's only caused by inhaling this bacterium, Legionella pneumophila, from water when it's aerosolized. Imagine if you're washing your hands quite vigorously, there's a little bit of water particles that will go into the air, and sometimes some of those will have bacteria on them. So then in that case, you may breathe them in and it can cause an infection. Now, in 2015, scientists uncovered that there was a contamination in the water of both lead and other heavy metal sources. And in October of that year, they switched back to the original source, Lake Huron. You can see over on the right-hand side is a map of the city of Flint, and the green dots represent places where Legionella infections occurred. So the PNAS article more closely tied the contamination issue of the water with Legionnaires disease. And what they found was that although both reports, I should say, are from the same groups of people. And in this PNAS report, they were able to tie the requirement for chlorine to disinfect the water and the amount of chlorine that was necessary to decrease the probability of Legionnaires disease was much higher in the Flint River source. So here we have pre-switch, that's when it was Lake Huron, and then post-switch, these red dots up here, that's when they were using the Flint River water. And then they switched back to Lake Huron, and that's those blue triangles. You can see that the amount of chlorine that was necessary to decrease the probability of a Legionnaires disease case was much higher when they were using the Flint River water source. And that's in part because of those particles of iron and other organic matter that was found in the Flint River water. These particles can react with the chlorine and change it to a form that is no longer able to be effective in disinfecting that water. The iron and other pieces of organic matter are also growth promoters for Legionella pneumophila, and so it could promote some of that bacterial growth and perhaps have a higher concentration of the bacteria, in addition to not killing the bacteria because the chlorine levels are decreased. What the researchers found was that the odds of a Flint neighborhood reporting a case of disease increased by a factor of 7.3 during Flint River water use. This is a very important finding, not only because of the number of people that were affected and the community's ability to have clean water. This is shared by many, many people, both researchers and Michigan residents, reporters, and this also has important implications in a case against the former Michigan Department of Health and Human Services Director, Nick Lyon. So the lead researcher, Michelle Swanson, is quoted as saying that it was the change in water source that caused this Legionnaires outbreak. And that's an important tie because this former director is now on trial for involuntary manslaughter connected to some of these deaths. And that's going to be this piece of evidence from these different journals is going to play an important role in determining whether this former director is charged or not. All right, we're going to move forward and talk about the other part of the water infrastructure, which is another MBio report titled Genomic Analysis of Hospital Plumbing Reveals Diverse Reservoir of Bacterial Plasmids Conferring Carbapenem Resistance. So the take-home message from this report is that bacteria swap drug resistance genes in hospital plumbing. This is not a good piece of news. So backing up, we all know that antibiotic resistance is a big problem, with the most recent numbers citing 23,000 people in the United States that die of these resistant infections each year. Now, this number is starting to get a little old, and I can only imagine will be increasing the next time that we are able to get some good surveillance numbers. So where do these clinical isolates come from? They can be transmitted from person to person. They can be transmitted within the community, but the isolates themselves, those bacterial species, often will pass those pieces of genetic material around on mobile pieces of DNA, things called plasmids or transposons. Those are pieces of DNA that bacteria can share in order to confer resistance in other species. And I have on the right-hand side a picture of an electron micrograph. This is one bacterial species sharing a piece of DNA with another. So what the researchers at an NIH hospital wanted to do was to get a sampling of exactly where are these drug resistance genes being shared. And so to do that, they used whole genome sequencing, looking at over 700 samples taken from a number of sources. They were particularly concentrating on carbapenem-resistant Enterobacteriaceae. These are real bad bugs such as E. coli and Klebsiella species that can cause a number of both localized and systemic infections. And they were looking at environmental samples. They were looking in the water infrastructure system at the water drains. They were looking within the plumbing, the sinks, which have previously been shown to be reservoirs of bacteria, as well as surveying both the high-risk patients and a monthly surveillance of all patients. And they made, in their report, they made several lovely infographics that helped to summarize the large amount of data that they accumulated. So in this particular figure, they have every isolate that they found that had a particular carbapenemase represented with a single square. And the different colors represent where they found it. So all the patient isolates are here in black. The red represents the wastewater sampling. This gray is the housekeeping equipment, things like mops or buckets that may be used to clean contaminated areas. The green here is the housekeeping closet drain. So you can imagine after using that housekeeping equipment, they need to dump the water somewhere. Then they also looked at high touch surfaces. So those are areas specifically like doorknobs or the keyboards of common use computers, things that might transmit those different contaminated sources. And then the blue here are the sinks that they looked at, and the yellow is the wastewater piping. And so you can see the relationship between different isolates represented by these arcs. The orange arcs here represent the sequences that are very, very similar between the patient and environmental samples. So you can see that there are certain connections between patient isolates and the wastewater pipes. A lot of connection between patients and the sinks. And then you also have these smaller blue arcs, which show environmental relationships. So those are sequences that are shared between different environmental isolates. And in investigating some of these environmental isolates a little bit further, they were able to determine the exact mechanism that one of these new isolates was generated. And they demonstrated this in another infographic. So the timeline goes that in February of 2011, an Enterobacter species that was carrying two different plasmids that had carbopentamases on them was cultured from one patient. This isolate may have colonized a sink in the patient's room. They didn't determine that, but they did see that it was in a patient. In the subsequent year, in January of 2012, a Klebsiella organism, an isolate, was found in another patient. And in this case, it also had a carbapenemase, but it was on a completely different plasmid. And again, this may have also contaminated the sink. The plasmid from this Klebsiella patient was transferred within this sink to the Enterobacter species, creating a third isolate. So because this particular Enterobacter has three different plasmids rather than two, it makes it a different isolate. And the three different species, the three different isolates are distinguishable because we have this very high-powered whole genome sequencing able to determine the exact DNA particles that are carried by all three of these different bacterial types or species or isolates. And so this has some serious consequences for the bacteria that might just be hanging around in the sinks. They're not necessarily infecting people, but they are still swapping some of these resistant determinants that could cause a really serious outcome if that new isolate is able to cause infection, especially in a hospital where there's a lot of very vulnerable patients who might be immunocompromised to begin with. And so this did have a pretty wide spread sharing both on social media and also was picked up by a number of news outlets. There were several co-lead authors, and one of them, Karen Frank, spoke with the Los Angeles Times and ABC News, or NBC News, excuse me. And although these are very serious in terms of hospital infection control, Frank did do a good job of trying to allay any fears for personal drinking water. So although there were these drug-resistant isolates that were found in the plumbing system, all of the drinking water that comes through our taps is something that should be treated at a separate facility before being shipped back. And that treatment should eliminate the drug-resistant bacteria. So people should not fear their water that they might be getting some of these different drug-resistant bugs. Frank said that we are not trying to send a scary message to people in their kitchens, but it does have these results will have some important implications in the ways that hospitals may dispose of their water or treat their piping. Okay, we're going to move on to the next article, which again was an MBio article titled Emergence of an Extensively Drug-Resistant Salmonella enterica serovar typhi Clone, Harboring a Promiscuous Plasmid Encoding Resistance to Fluoroquinolones and Third-Generation Cephalosporins. Quite a mouthful. The take-home message from this is that an extremely drug-resistant typhoid fever may halt first-line drug efficacy. So typhoid fever is a disease that's caused by Salmonella enterica serovar typhi, or S. typhi, and is a problem more in the developing world. So this is a map in the right-hand side, highlights the countries where you see higher incidence of typhoid fever, in part because the United States has a good healthcare system and can treat people who become sick with this disease, and also in part because we have a very good water treatment system. This is not a disease that is highly prevalent in the United States, but you see sub-Saharan Africa, parts of Asia have quite a high incidence of this disease. And this may sound familiar to you. This was one of the pieces of news that was sent out in the ASM Weekly News Digest. This is an email that ASM staff and ASM members can sign up to receive. You do have to opt in. So if you're interested in getting a news summary every week of the hot news in microbial sciences, we'll include a link below where you can sign up and get that in your inbox as well. We're going to talk a little bit more about what they actually found, which was an addition to some of the already present resistance genes carried by S. typhi. So S. typhi already had several different chromosomally encoded genes that allowed it to be resistant to certain drugs. So there's already a chloramphenicol resistance gene, an amoxicillin or ampicillin resistance gene, a trimethoprim or sulfamethadol resistance gene, several different resistance genes, and a ciprofloxacin resistance gene. All of these are on the chromosome. So that means that it's passed within the bacterium itself, but it's harder for those to be moved by horizontal gene transfer, such as conjugation or sharing of plasmids, right? But in this outbreak in Pakistan, they found a plasmid, which they called P60006, which contained two new resistance genes to quinolones and to extended spectrum beta-lactamase, as well as a locus that allows this plasmid to transmit itself to other Salmonella typhi and paratyphi serovars, meaning that they too could become resistant to these important drugs. And what the take-home message from all of these different resistance determinants is that only azithromycin remains as an effective oral antibiotic option in these developing countries. Now, should this isolate make its way to the United States or the UK, there are second-line drugs that you can treat people with, but those are much more expensive, and in a place with a much lower per capita and a GDP and a much lower, a still weak, in some cases, health infrastructure, that's not going to be cost effective to treat outbreaks in the developing world. And so what that means is that in some places, because of access issues, this may soon become a drug-resistant infection. That is, with only one more resistance mutation, azithromycin will no longer be effective. Fortunately, there is another option. Recently, a typhoid vaccine has been approved. And so what this emphasizes, as the lead author Elizabeth Clem said to The Guardian, is that the increased levels of drug resistance add an urgency to the call for vaccination. So being able to prevent disease is going to become much more important because we won't be able to treat the disease. This was echoed by Charlie Weller, who is the head of vaccines at the Wellcome Trust, which funds a lot of biomedical research, who also said that focusing on prevention is going to be the necessary course. And so this news was highlighted by researchers and journalists alike, and is something that is going to play a big role in global health going forward. All right. Those are all relatively negative pieces of data having to do with outbreaks, but I do have a very promising study from Nature Microbiology titled Culture-Independent Discovery of the Malacidins as Calcium-Dependent Antibiotics with Activity Against Multidrug-Resistant Gram-Positive Pathogens. And so the take-home message from here is that a new class of calcium-dependent antibiotics was found in soil. And soil seems like a natural place to look for antibiotics. A lot of the antibiotics that we have in the clinic now are produced by microbes. They were first discovered in microbes, such as the compound produced by Streptomyces griseus, which fights tuberculosis, and many others. These antibiotics are produced by microbes who are trying to kill or at least inhibit their neighbors from accessing some of the scarce nutrients in the soil. And so what this group did was to take DNA from the soil. They collected the DNA. They did not grow or culture the microbes themselves, and looked for genes that looked similar to others that generate antibiotics. From that, they were able to make a library and clone that pieces of that library into model organisms, both E. coli and Saccharomyces cerevisiae. They grew large quantities of the different clones, and then tested the products for their ability to inhibit different organisms. What they discovered was a compound called malicidin, which they were able to determine binds to the lipid 2 component in a calcium-dependent manner. So this is a schematic from that same group demonstrating how malicidin works against against certain bacterial organisms. It's comparing it to other similar compounds that are similar in their structure, not necessarily in their mechanism of action. You have to see here daptomycin that actually works against the cell membrane. And so what we have here is a very, very close-up view of the cell membrane. This is the extracellular area that's going to have the cell wall material, and then the inside of the bacterial cell. And they discovered this malicidin works best against gram-positive organisms. It inhibits the growth of the cell wall, and the cell wall is much larger in these gram-positive organisms. So malicidin binds to lipid 2, which is the same target of the drug vancomycin. But although they found that they both bind to lipid 2, they likely bind in different ways because malicidin is still active against organisms that are vancomycin resistant. Over here on the right-hand side, I have a table that the authors published showing the efficacy of malicidin and the minimal inhibitory concentration, which is quite low against many of these different gram-positive organisms, those that have that really thick cell wall. You can see that there's not a great deal of efficacy against gram-negative organisms, such as E. coli here, or fungi, such as Candida albicans, shown here. But that even resistant isolates that are not sensitive to vancomycin are sensitive, and indeed sensitive, to this malicidin. So that's very promising. We discuss antibiotic resistance all the time on these microbial minutes and the need for new antibiotics. And so this is really a very promising finding. And that's one of the reasons why it was highlighted by people on social media as well as the news. This was covered in the Washington Post and BBC, where the lead researcher, Sean Brady, said that our idea is that there's reservoir of antibiotics out in the environment. It hasn't been accessed. And so I believe that the Brady lab will continue to mine some of these genes that they've been able to find in the soil to look for more that may produce antibiotics. But it's important to temper our expectations. This is not something that will be clinically relevant in the next year or two. It is a long, arduous road from the initial discovery of an antibiotic to a clinically used entity, as Brady himself emphasized. But because this is such a dire need in the clinic, it was highlighted by a number of research institutions, people associated with different medical funding, and even the NIH head himself, Francis Collin, highlighted this in his director's blog and why it is important. And so we have one more item that I want to cover that wasn't mentioned in the beginning, and that is the scaling back of CDC funding in foreign countries. This is a story that's been unfolding slowly as different rumors have been spread. The Atlantic had a very nice summary of what has been said and when, and it looks as though it's been confirmed, since it's been reported by both the Wall Street Journal and then the Washington Post confirmed that funding for CDC outlets in other countries is going to be severely decreased starting in September of 2018. So that's this year. And this is, of course, something that is important because the CDC, although it is a United States entity, a United States institution, helps with both surveillance and with response during outbreaks. The most recent large-scale example would be the Ebola epidemic of 2014 to 2016 that occurred in West Africa. The CDC was very important in helping to build some of the infrastructure to set up labs for both testing and diagnostics as well as treatment. And this is something that will have, again, a lot of impact on global health because diseases do not respect national borders. And the ability of diseases such as Ebola or influenza, as we highlighted earlier, that can be carried in animals, or you would imagine any type of contaminated water that may cross between countries, could carry that contamination as well. And this is something that could potentially have a very large negative impact on transmission of disease globally. All right, so we've talked about a number of different things that have happened in the last couple of weeks. Talked about the avian influenza, the new avian influenza strain that has jumped into people, Legionnaires disease outbreak tied to Flint water quality. We discussed how bacteria can swap drug resistance genes in hospital plumbing, and the new extremely drug-resistant typhoid fever that may halt first-line drug efficacy. Finally, we talked about a new class of calcium-dependent antibiotics that were discovered in soil. I'd like to thank you for attending our Microbial Minutes sessions. Remember, we have these sessions every couple of weeks, trying to summarize what's hot in the microbial sciences and talk about the primary literature from which it comes. If you're interested in more of these sessions, please subscribe and hit like so that more people can find us. We'll talk to you next time in another couple weeks with our next Microbial Minutes session.